Okay, so for my thesis work, I'll be explaining my investigation of the expression patterns of tumor suppressor BRD2B in early vertebrate development. And BRD2B is a paralog of zebrafish BRD2, or bromodomain containing 2, which belongs to the Bet family of proteins, which I'll explain a little bit later on. And BRD2B derives from genome duplication events from BRD2. And because gene duplication events can confer overlapping as well as novel functionality, studying BRD2B provides an implication for studying the evolution of gene function as a result of gene duplication events. And because BRD2B is unique to the teleos lineage, of which zebrafish belongs to and harbors this unique paralog, I'm using this species as model organism for studying BRD2B. So as you guys can see here in this phylogenetic tree of the bromodomain family within the zebrafish organism, BRD2B is closely related to BRD2A from the gene duplication events, bringing about the two paralogs. And BRD2B belongs to the Bet family of proteins, which are critically involved in cell cycle control, transcription, and development. They're characterized as epigenetic co-regulators that bind acetylate histones and act as scaffolds for ensuing chromatin remodeling complexes, as well as act as bridges for transcription factors to basal-level transcription machinery. And in this way, they've been shown to be involved in cell death, differentiation, and division. And they achieve this functionality through characteristic domains, one of which is a bromodomain, which is used to bind acetylate histones, as well as the other one is the ET domain, which is putatively known for protein-protein interactions, presumably within the scaffolding functionality of the Bet proteins. And previous work on BRD2B has shown that it is a maternal transcript heavily enriched in developing oocytes in zebrafish, as well as early embryogenesis in embryos. And RNA in situ hybridization of both BRD2B and BRD2 has shown in stage 1 and 2 oocytes that both paralogs have similar overlapping expression domains within the oocytes, but shown here in late stage oogenesis in stage 3, the RNA in situ hybridization for BRD2 is shown in panel A. And what we see here is that the RNA is heavily enriched around the cortical periphery or the outside interior of the developing oocyte. In contrast to that, we see that the RNA in situ hybridization patterns for BRD2B vastly differ from that of BRD2, in that the panel here for the stage 3 oocyte stained for the RNA for BRD2B, we see that it's highly localized around a special follicle cell denoted the micropyle. And at this follicle cell, that is the demarcation of the future animal pole for developing egg polarity in the oocyte. As well as we see that in developing embryos, both BRD2 and BRD2B overlap in the central nervous system of the embryo shown here in panel C from a dorsal side, as well as they have minute small differences in expression. And if in fact function is dictated by structure, this could be a situation where BRD2B and BRD2 are somehow functioning in relation to each other to establish early embryogenesis for central nervous system development, as well as oogenesis and egg polarity required for oocyte development. And to further characterize this functionality between BRD2 and BRD2B, the BRD2 gene is predicted to potentially encode 2 RNA alternate transcripts, one of which was previously cloned and identified through screening a cDNA library of embryos, which is shown up here, as well as on the bottom is the purely predicted RNA transcript shown through bioinformatics. And what's very interesting is that within the transcript of the predicted RNA transcript from bioinformatics, we see that the translated region, bigger section right here, contains characteristic Bet domains. We have the bromodomains in red, 2 of them, a nuclear localization signal in the stripe box, an ET domain shown in yellow, and that's, again, characteristic of the Bet family through the bromodomains and the ET domain. However, in striking contrast, we see that within the previously cloned transcript, that within the translated region, it only encodes a single bromodomain, which is atypical of Bet proteins. And further analysis reveals that within the last coding exon of the transcript, when translated, it introduces a premature stop codon, thereby truncating the translated protein, accounting for the single bromodomain. And this intronic region is conserved within this transcript and is within the exonic region in the BRD2B gene locus. And if in fact this intronic region corresponds to a true transcript and is not a byproduct of the cloning process. This could be an antagonistic functionality between BRD2 and BRD2B between their hypothesized interactions. Because BRD2B has been shown to be a tumor suppressor and BRD2 has been shown to be a proto-oncogene, if in fact we have modulation of different RNAs and translated proteins, we could have situations where you have cargoless single bromodomains translocating to nucleus independent of nuclear localization like traditional pathways, interfering with the traditional chromatin binding of BRD2. And so given all this information, my experimental aims, I want to identify the predicted and cloned BRD2B RNA and protein isoforms in staged oocytes and embryos with reverse transcription polymerase chain reaction and Western blot analysis, as well as verify that the in vivo transcript, whether in fact that intronic region is actually expressed in a true transcript or is merely some sort of artifact of the cloning process, as well as examine the expression of BRD2B protein isoforms throughout oogenesis and embryogenesis for potential functional insight and differential expression patterns based on temporal development. And all these experimental aims coalesce into my hypothesis that the BRD2B gene encodes multiple RNA and protein isoforms that are differentially expressed in oocytes and embryos. So for my RT-PCR method, I isolated RNA from stage 1 through 4 oocytes, purified the RNA with a column with DNase treatment, and reverse transcribed the RNA to cDNA. Similarly, I also isolated the RNA from stage 4, 24 to 48-hour developmental time points of embryos, column purified the RNA with, as well as used DNase, and reverse transcribed that RNA to cDNA. And from there, I made short oligonucleotide PCR primers spanning specific exon regions within the predicted RNA transcript as the cDNA. And as you guys can see here in this top RNA transcript, these exon-exon spanning primers shown in the yellow arrows correspond to exonic regions previously unconfirmed by bioinformatics databases, as well as within the previously cloned sequence, the RNA transcript shown below, I also designed primers spanning the exon-intron junction of the conserved intronic region unique to the smaller RNA transcript. And to tease apart any significant results that may correspond to that PCR product spanning the exon-intron junction, I included DNase treatment to get rid of any potential genomic contamination that would give me a result that may be a false positive, as well as I made a positive control spanning an intronic region unique to the BRD2B gene locus that if in fact there was any genomic contamination would give me a product. And for looking at the protein expression of BRD2B, I employed Western blot analysis by staging oocytes through stages 1, 2, 3, and 4, as well as staging embryos from 4 24-hour and 48-hour developmental time points, submitting them through SDS page and Western blot analysis on a per oocyte and embryo basis, and verifying any peptide bands with a peptide competition assay in which I incubated the primary BRD2B binding antibody with its binding epitope to ensure the specificity of the antibody. And for the RT-PCR results, what you're seeing here is agarose gels run from the PCR results through stages 1, 2, 3, and 4 of oocytes. And most interesting is that through every stage of oogenesis, we have expression of the previously unconfirmed expressed exonic regions. And looking at the embryo stages of stage 4, 24, and 48 hours, we have the expression of the previously cloned RNA transcript with the inclusive RNA transcript, intronic region. And that small intronic region shows to be expressed between the 4 and 24-hour developmental time point of the developing embryo. And because during that time point, the segmentation is almost completed at 24 developmental time point, this could be a tissue-specific transcript being accumulated and expressed from segmentation on in that specific tissue expression. And not only this, this confirms that in fact that this previously cloned transcript corresponds to a true in vivo transcript, and that's not an artifact of the cloning process. And to further verify the specificity and that these PCR products conform the desired products I designed, I sequenced all of them, and they all map back to the transcripts of which I designed the PCR products against. And my Western blot results also give some key evidence. Shown here is the Western blot results for two separate experiments. Left was the embryos, as well as the right was the staged oocytes. And here what you're seeing is two different potential protein isoforms being shown in late stage oogenesis in stage 3 and 4, as well as the 24 to 48 hour developmental time points show again a similar expression of the two protein classes. But what's most striking is that the primary antibody was made against only the predicted RNA transcript, but not the truncated RNA transcript. So these protein isoforms are potentially picking up that protein that's being translated. But when you translate that protein, look at its molecular weight, it's roughly around 90 kilodaltons. And here we're getting proteins around 37 to 50 kilodaltons, and between 150 and 250 kilodaltons, which greatly differs from the predicted molecular weight. And given the fact that these proteins were submitted to reducing conditions with SDS page and BME, this could be a situation where we have irreducible complexing, either through chromatin or some sort of protein processing through post-translational modification, accounting for the high molecular weight bands that was unexpected. And to further confirm that these bands do correspond to the BR2B binding epitope the antibody was designed against, my protein peptide assay, peptide competition assay, is shown on the right here. On the top we have the MENCODE stain, which shows protein expression on the nitrocellulose membrane prior to incubation with the antibody that was priorly incubated with the binding epitope, and all the signals disappear, giving evidence that the antibody is specifically binding to its designed epitope that is corresponding to BRD2B. And what we see too is that at the 4-hour post-fertilization developmental time point of embryos, there appears to be lacking any sort of protein expression, giving suggestion that this could be a situation where you have degradation of maternal proteins that are upregulated from zygotic genome expression between the 4 and 24-hour developmental time point, playing critical roles in embryogenesis and oogenesis. And so given these results, my conclusions are that both the intronic inclusive BRD2B RNA transcript and unconfirmed exonic regions are expressed in vivo. And the larger RNA is a maternal and zygotic transcript, while the intron-inclusive RNA is likely a zygotic transcript presented during segmentation of central nervous system development. And that the maternal BRD2B protein appears to be degraded, and that the zygotic BRD2B protein accumulates between the 24 and 4-hour post-fertilization developmental time points. And finally, that these proteins are somehow post-translationally modified or potentially tightly bound to chromatin, not being broken up by reducing conditions. And so I'd like to thank the Villanova University Department of Biology, members of the D. Benedetto Lab, Dr. D. Benedetto, the Wyckoff Lab, the Jackman Lab, and I'll be taking questions now.